Okay, I think now it works. I hope. Okay, yes, so it does work. I can see myself. I can hear myself. Hi, hello, everyone. Ooh, this was now a little bit a very stressful couple of seconds here because I could not see the stream go live. Hi, hello, everyone again. I'm Oliver, Microbehunter here, and yeah, I think we can get started again with another Saturday live stream. It's great that you're joining me again here. I see that many people already started to post in the chat. I've prepared again a couple of topics for today, and that what you can already see here. That's actually a live image. Yeah, what you see over here. That is actually these soy sauce crystals, and they look kind of pretty. So we're going to be playing around with those here as well. And however, that's not the only thing because a few weeks ago I changed the format around, and for this reason I decided to split the live stream into several topics, and that's what I've kind of prepared for today. First a couple of minutes editorial, 15 minutes. So these are the minutes. That's where I'm going to say hi to around the world. I'm also going to show you some pictures that were sent to me. I want to talk a little bit, make some crystals. This has been also a request a little bit to put salt and soy sauce under the microscope. It's a leaf imprint that I've made, which actually worked quite nicely. I mean, it's not the first time that I'm doing these things here, but every time when I do that, there are a few small variations. Elk meat I've got that I would like to show you. And then I want to do a little experiment called density gradient separation, which I've not tried yet, but I want to try it out live with you to see if this works out as a method to isolate microplastics. Okay, I'm not going to say more to that, only when we're there. And then a Q&A session. So if you have any questions, any comments or so, please feel free to post them in the chat section. Please also indicate at Microbehunter or at Oliver so that I know that the question is directed to me. And I'll spend the last 15 minutes here. Yes, hello from Vietnam, from Belgium, North Carolina, from the Netherlands. Okay. Yeah, hello from Upper Austria. Hello, hello back. Yes, hello from Florida. Yeah, from northern France, from Ireland. Okay, yeah, and what I would like to do is I want to show you a couple of pictures here because someone sent me, also a fellow hobby microscopist, sent me some very nice pictures using basic epifluorescence. And I don't know if you've joined in now in the chat, but if you have, then please, please feel free to comment on those. I think that's quite nice. These are the pictures of a spider, and very thankfully, he sent them to me to show to you. And I would like to simply show this to you, those pictures. And I don't know, I hope that you've joined in the stream. If you have, then maybe you can also comment on those. This is basically the picture of a spider using UV light coming from the top. So it's using an inverted microscope, basically one with the optics on the bottom, and then there is ultraviolet light and different colored light coming from the top, and then the specimen starts to glow and fluoresce. And this is actually a thing that I have not seen very commonly done yet in the field of hobby microscopy. First of all, because UV microscopes are expensive, but maybe it's also possible to use another source for the ultraviolet light. And it's quite nice here in the spider because you can actually see that depending on the wavelength and on the color, the different parts of the spider start to fluoresce and to glow. So thank you very much for these images that I'm able to share them with you. Over here, I've also got a description for each one of the images. I can also show them to you at all of the wavelengths are indicated, but essentially there is usually always a blue light or UV light which kind of excites the different pigments in the spider, and then it starts to glow back in a different color. So this is actually quite a nice thing also to experiment. Now what I have also got over here is this is also a UV flashlight. Which yeah, and I'm also going to, I mean, I've this is also on my list to experiment a little bit with also with epi illumination using ultraviolet light, right? So this is something that is quite quite nice to see, and I think the pictures look quite nice. You see, there's always also in the corner a description, yeah, it's a technical description here, yeah. So thank you very much, yeah. Yeah, thank you very much for those pictures. I think it's quite quite nice yeah, to see different lighting and illumination techniques that yeah, even I am not doing, right? Yeah, and yeah, so it's UV or short wavelength light from the top or bottom and then filtered with a long pass only to detect the low wavelength. Yes, I'm going to, if you're interested in the technical details, I'm just going to quickly show you this here. That's basically what I was sent. And yeah, here is a description. Yeah. Yeah, it was also in German, but I translated this. So there's an excitation wavelength. Yeah. And then the detection, that's basically what, yeah. Yeah, CT. I think that might be you. If you are the one who sent them to me, thank you very much. It's UV or short wavelength light from the top or bottom and then filtered with a long pass only to detect the lower wavelengths, which is this one here, the 425 nanometers. Yeah, and RLD is a reflected light detector. Right, very, very nice. Yeah, it's pretty cool. So you basically shine in, you use one wavelength to illuminate it, and then you detect it with from a different wavelength. Yeah, so this is quite nice. Yeah, so editorial, another usually 15 minutes. As I said, a couple of other things. You've noticed that yeah, if you want to send me also pictures or short video clips to share, then please do so. I'm very happy to present them. Yeah. So what else is there? I need to make an important, semi-important announcement, and that is that next week there will not be a live stream because I will not be around. Okay, just just for your info, I will yeah, just so that you don't expect a live stream. Yeah, fairy shrimps coming soon. Very nice. Okay, yeah. So. Another thing that I just wanted to mention, it's a little bit of I don't know, this is an idea that I have, and I think this is, I don't know how to do that really, right? A comment from ever smaller from Jesse. But what will I do without Oliver for a whole 2 weeks? What I will do is, if you probably noticed, I have increased the upload frequency of videos on this YouTube channel here. A few weeks ago, I changed around the format a little bit. I'm now using this Microbehunter Microscopy, this YouTube channel that you're watching right now, to do more the hands-on microscopy live streams and videos. And the main channel Microbehunter, I want to do. I'm still struggling a little bit with that. I'll be honest with you. I want to do more type of explainer videos. I just finished one today, an explainer video about spontaneous combustions and how microorganisms contribute to that, but I was not happy with it. Right, so I'm redoing that video. So you see that for the other channel, I'm still kind of working a little bit of finding a proper workflow. But this channel here, I want to keep you a little bit more updated, and I actually hope to be able to, and I don't know if this is going to work out, I actually hope to be able to, because I'm on a holiday next week, maybe to do post a couple of videos from my holiday place, and that's one of the reasons why I took along or bought myself this USB camera here, and then maybe I can, I don't know, show you some, I don't know, rock samples that I find on the side of the road. I don't know, we're gonna see. Okay, yeah, so yeah. Okay, yeah, so this is yes, another idea that I have, and this is something that honestly I don't know if this is just a vision for the future and I'm not able to realize this. when I was still at university. This is I don't know. I graduated from university back in 1998. That's quite several years ago already. When I graduated from university with I studied microbiology. I went to a couple of conferences, really nice ones, scientific conferences. So we presented a poster with our scientific research and so on. And I was kind of wondering if it would be kind of nice to have conferences or meetups or so, for hobby microscopists. Yeah, so this would be something nice, but I know that it might not be quite a realistic thing to do. But I don't know. It's just something that I would kind of wish for, that somehow there would be the possibility of personal meetups with other fellow hobby microscopists. Live streams like this are very nice also for me, because of course I see that there's a community there. But I'm of course obviously only able to see text comments only. I mean, I'm really happy that this is possible. But of course, it's a different thing if you can actually personally meet up people as well. So I have been thinking about, and I don't know if this is actually something which is a good idea or not. Maybe, I don't know, having some kind of a virtual conferences, Zoom meetings where different people can present different things. I have no idea. You get the idea, right? So this is a little bit something that's been going all around in my mind for some time. So let's start a little bit with the actual microscopy itself, and with the soy sauce and the salt crystals, because this was actually a request. Yeah. Jitsi, yes, exactly. It is, but it would be Jitsi is a video conferencing platform which we've already tried out several months ago. So where is this? Here we are. So very briefly, in order, if you want to make so you see it over here on top. So all I have done is I've taken some soy sauce. Here it is. Directly from the bottle and spread it over a microscope slide and then let it dry. And because drying takes so much time, I used this thing here, that's my coffee warmer, to speed up the drying process. And this is basically over here, this is what we get, right? Let's have a look at it first a little bit, and then let's add a little bit more soy sauce. So, and this was also a question a little bit. CT. Okay, yes, I'm just reading the comments. You see colors. This is because of DIC. So what you are going to see is you're going to see it more like this. So that's a bright field. These are the natural colors, of the crystals. And I think the patterns, let's put on the color back again. I think the patterns look quite nice. I suppose that's not only salt, but all of the other substances that are in there. Usually soy sauce being a natural product, fermented, I think, as well, probably contains countless substances. But what I would like to do, that's the second slide here, is I would like to add a little bit more soy sauce here, and let's have a look on how the crystals actually melt away. And then we should slowly see the crystals reforming again. Right, because as the soy sauce evaporates and dries up, essentially the crystals start to reform again. So what I'm doing right now is I'm just going to add here on the side a little bit of soy sauce, which you cannot see because it's off center. And here we go. By the way, no cover glass, and that's why I'm using a low magnification. And I wonder what those little circles are that you see. I wonder, are these some fat droplets, or I don't know. Yeah. So that's quite, Yeah, so you see that, The crystals do not quite dissolve as quickly as I expected it, and that is of course because the soy sauce now seems to go into saturation, and then of course the dissolving process also slows down. Yeah, so probably oil. Yeah, it looks those round droplets structures that you see. If something is really that spherical and that round over there, those yeah, then this is actually a very strong indicator that we're indeed talking looking here at some kind of an emulsion here of oil and water. Because what happens is that oil, being hydrophobic, does not want to be in contact with water, and therefore it will go into a circle or a sphere because this has the least surface area to be exposed to the water. Yeah, so you can actually see already it's slowly starting to slow down. Yeah, and this is something that can often be observed, that even though there is liquid still present, the crystals stop dissolving or slow down because it's already saturated. Yeah, and when it's saturated, then basically the whole process stops. Yeah. And what we could do is I'm going to try to follow. I'm just going to spread it around a little bit here. Just going to destroy it. Okay, let's go down with the magnification here. Yeah, and those fragments, as essentially as it evaporates again, it will act as so-called crystallization seeds, and then crystallization will start again from all of those fragments, and this actually looks quite pretty. And because I'm impatient, or we're all very impatient, what I'm going to do is I'm going to speed up the whole process by simply putting the slide on my coffee warmer. Okay, this speeds up the evaporation, of course, and yeah, it says 55 degrees. And a lot depends also on the ambient, of course, on the ambient, not only temperature but also humidity. Because if the air is very dry, then of course the soy sauce is going to dry up much faster and quicker, and then you can actually see the crystallization process. Let me see. It's actually fairly hot. The 55 degrees. I mean, I think this is not quite accurate. Yeah, this is definitely hotter than 55. Huh? So let's. I think that's cool enough. Let's go back here, and yeah, you see that. Look, yeah, it's all of those patches that you see there. This is basically where crystallization has started. Let's go up. Yeah. And what I have done also, it's already several months ago, I have filmed the whole thing for, I don't know, quite some time. Yeah, here we actually see how it's growing. Let's go here. Yeah, and then I did a time lapse. But this is actually, it's nice. Here we can actually see how, yeah, the crystals are forming. Look at the tip, yeah. So that's now basically in real time. And if you kind of take your time with 10 or 20 minutes, and then when you film that and then you speed it up, then of course, it's going to be much more impressive because it's much faster. What's also going to happen, however, and sometimes it does stop, is that it's not only when the liquid completely evaporates, but sometimes, of course, all of the salt and whatever substances that are here will deposit on the crystal, and then, of course, it takes time for new substances to diffuse towards the crystal, and sometimes it will stop after some time if all of the substances have already been used up. Yeah. So I'm going to just use this. Okay, see, it seems that it kind of stopped now. So let's go down a little bit and try to find another place. So in the meantime, I'm just going to read a couple of comments as well. And then we're going to have a look at some salt crystals, which again look different. Maybe use high magnification with a cover glass and add alcohol or water at the side to induce crystallization. Yeah, the thing is the following. The reason, I mean, of course I can try that. The reason why I did not use a cover glass is because the cover glass on top will actually limit the evaporation of water. then it's only able to evaporate from the side, and then I've found out that the crystallization process, while it still happens, is much slower. And that's why I usually, when I observe crystals, because they're at low magnification anyway, I usually do that without a cover glass. Yeah? But it doesn't mean that you cannot do it the other way around as well. Yeah? Can we do the same thing? You know what? Let's try it. I don't know. I don't know if alcohol or to what extent alcohol actually also works. Okay, so what I'm going to do is I'm going to drop alcohol over it. Yeah. But you see that over here, it looks like that the whole process has already stopped, which is a pity. But of course, we can now try different solvents. Yeah. So, but now we cannot see the process as it happens. Yeah. I assume most of what we are seeing crystallized is simply salt. It could be, but I think there are also quite a few other substances in soy sauce. What are certain amino acids? I think those taste enhancers, glutamic acid, glutamate. I'm guessing right now that actually can also crystallize out. Yeah, and I have also seen once, actually today in the afternoon when I did a trial run here, that there were actually two different type of crystals forming, and it could be that maybe different substances crystallized out. Because if, for example, sodium chloride salt starts to crystallize out, then of course only sodium and chloride will start to accumulate on the crystal and not other substances. So actually, crystallization has been used also in chemistry to purify substances, because by forming a crystal, then you have the pure substance. Okay, but as requested, here, let's again the other slide. Let's do the following. Let's go down to the lower magnification, and let's simply try alcohol now, because it's a different solvent, and I don't know if it works or not. And you know, the only thing I need now is a container for the alcohol. Just a sec. Oh, here it is. So I'm just gonna add a little bit of. I've always got a little alcohol bottle here, because I use alcohol also for removing certain stains and yeah. So that's alcohol, and here. So up, I bumped my microphone. I'm just gonna show you here what I'm doing. I'm using a new tip. Yeah, and now I'm transferring this on the slide, and let's see what happens. Let's take, I don't know where it's kind of dense, maybe here I don't know. Let's see what happens. Because alcohol has a much lower surface tension. I mean, something is happening. Let's add a little more. No, I ran out of. Huh. Yeah, no, you see? A little bit as interesting, a little bit maybe as expected. The alcohol does not dissolve the crystals, because the alcohol is of course more hydrophobic and the crystals are hydrophilic. I put it directly on top. No, the whole slide is flooded actually. You can actually see that there is some flowing. Yeah, the liquid is flowing, but here's something going on here. I mean, it does dissolve a little bit, but this is strange. Let's open it. I mean, it does seem to. It almost looks like if the fat is being dissolved out. Can this be? I mean, something is bleeding out here. Something is bleeding out here. Yeah, but it's kind of strange. Yeah, but the crystals themselves don't seem to be so much affected here. Interesting. Yeah. Yeah. I wonder what would happen if you put a drop of soy sauce into the alcohol rapid crystal. I have no idea. You see, I have no idea, so that's why we're gonna try it out. Okay, so just for your information, there is now a layer of alcohol over the slide because the alcohol's low surface tension causes it to spread a lot. The alcohol, of course, is also evaporating. So let's quickly add a little bit of soy sauce on here. You see that with the polarized light, so that's without polarization now, okay? And look what happens. Of course, the soy sauce containing a lot of water dissolves it. Yeah, interesting. Interesting that the alcohol didn't. Well, I mean, which doesn't. It's not entirely surprising because salt and alcohol also don't mix well. Yeah. That's yeah. Let's add again a little bit of alcohol. I have to change the tips every time. And it's this type of experimentation that sometimes is also quite fun. So look how the water and the alcohol mix and causes these patterns. That's because of the different refractive index. Salt, the water and alcohol have a different refractive index, and look what's going on. Wow, look at this. This is surface tension acting, yeah, because the alcohol destroys the surface tension, and yeah. And then you see the movement, yeah. It's not only here. Generally, when you have certain solid particles and you mix alcohol and water, you can see that there's always a swirly movement going on as the alcohol destroys the surface tension and then evaporates, and this causes a flow. Yeah. Yeah, this is a very nice example of the intersection between art and science and microscopy. Yeah. So let's do the following. How do you determine if a substance can make crystals or not? Well, you gotta try it out. I mean, generally, it's like this that certain substances crystallize easier than others, obviously. But you try to get a substance, you put it, and you try to dissolve it. So what I've got is a whole range of different substances. For example, aspirin. If you get the pure form, highly pure form, not the one pressed into tablets, but it's possible. This also dissolves I think it dissolves quite well in alcohol. Caffeine, if you get pure caffeine, also makes beautiful crystals. Vitamin C dissolves both in alcohol and in water. So there are plenty of things to experiment around with. Some crystals look nice, others don't look so nice. The soy sauce one actually belongs to the nicer ones. It's simply the patterns that are made. So yeah, hydrochloric acid. Wow, that would be interesting. Can you even crystallize that? Because actually HCl, hydrochloric acid, is needs to be in water in order to. Yeah. So and now what I have here is another thing, and you don't see you see this over here. I need to always occasionally when you make a mess, you have to clean it and. I'm just going to show you the following. Why not try this now? Yeah, I'm going to turn it off. Look, now you have to focus a little bit on the small, how do you say, picture on top. I'm just lowering this. Okay. And I'm opening the screws behind here. I'm taking this out. Here it is. And I just want to give the microscope a quick clean. Yeah, because any soy sauce or salt that has basically spilled over, yeah, I just wanna remove that. That's it. Yeah, shouldn't be a problem in any way, but yeah, let's put this back. Yeah, now you can actually see that the top camera, is not the fastest one. I'm aware of that. Yeah, it's always a little bit delayed. Yeah, and the frame rate is pretty bad. I'm aware of this issue. Unfortunately, I was not able to find another webcam that gives me that's so small and gives me also a wide angle. Yeah, so just yeah. So let's move this up again. Okay. I have betaine HCl capsules, which says it's pure without any additives. So I figure if I dissolve one, yeah, I think that's the whole point of this hobby, trial and error, right? And then we kind of learn, we learn from that. Yeah. Yeah, Wout says structural biology student here. I don't know about inorganics, but for proteins, making crystals is almost a science. Yes, I also heard that, especially when when you try to make crystals of proteins for X-ray analysis, for structural analysis. I heard it's for some proteins extremely difficult to crystallize them. Yeah. And you need to have a crystal in a pure form in order to actually get a proper X-ray done from that. So let's X-ray crystallography. That's how it's called. So what I've got here is all a slightly different thing here. I'll need this later as well. This here is saturated salt water, which I also made. So I've taken a lot of salt, some water, and I heated it up in the microwave, and you allow it to cool down, and you can see that it starts to form crystals all over the place because cold water is not able to hold as much salt. And when we put a drop on here, like this, then essentially we just wait. And we should see crystals forming. And in order to speed up this process a little bit more, because I'm a little bit impatient, we're just going to put it again on the hot plate. And as soon as the first crystals start to appear, I'm going to put it under the microscope. Salt crystals, basically, if you use polarized light, they do not give you colors, but they still look pretty. Yeah. So I'm just in the meantime. Yeah. Yeah. So it's a branch within biochemistry. It's all about the structure of biomolecules and how they give rise to function, structure and function. In biology, the function of a molecule, like a protein, a complex molecule, is determined by how it looks like, its structure. So it's this structure-function connection here. Okay. So you know what? I think the first crystals are forming already on the edge here. So let's have a look. Okay, I need to tighten this here. And scope. So this is again bright field. I always have to swing out the condenser if I want to get the full field of view. Yeah, and this is basically bright field, and what we can already see is those tiny square crystals forming. Let's add a little bit of color, because it's nice to have some color. Okay, like this, for example. And then the same thing, we can start, and watch how the crystals grow. Yeah. Now let's move the arrow out of the side. And so this is actually something that I have been doing in the past quite a lot. Is going back over 20 years, even before the digital age. I had an analog camera with film connected to my previous microscope, and I took pictures and I framed them, and I have some of them still hanging somewhere. So this basically means it's microscopy also is a possibility to do a little bit of art. Yeah. So, and what I've done is I've made a lot of these slides with all sorts of different substances, and then simply took pictures and on film, analog film. I got it developed, and then I got them pretty expensive at that time still. got them printed out in poster size. Yeah. Yeah, so the pyramids. The thing is yes, by not adding a cover glass, I'm also not restricting the crystallization process very much, right? So I'm just allowing it to crystallize freely. Yeah, yeah. So you get the idea. You know what? I would like to move on a little bit, and for this reason, I'm just going to very quickly and put it again on the hot plate. Just for a minute or so. And another important thing is that some substances are hygroscopic, which means they attract moisture. So it can indeed be that some substances they do not like to crystallize very easily because the humidity in the air is enough for it to not crystallize. Yeah. So that's also something that, So, yes, you see the crystals are now quite a bit larger. Yeah. Yeah. So yeah, very very nice. Also a possibility is, and this can be done more easily with sugar. You make a concentrated sugar solution and then you put a string into the sugar solution and then you watch the crystals grow on the string as the water slowly evaporates. Yeah. Yeah. Okay. Just some playful ideas. Also because I did get a comment the other day that somebody wanted to see some salt crystals, and I just wanted to say that salt is quite easy to do. Just make sure to make a concentrated salt solution so that you do not have to wait so long for the crystals to form. Let's move on. Okay, next topic. What's the next topic? Salt crystal, leaf imprint, totally different topic. Okay. Totally different. So we're jumping away from chemistry. We're going over more into biology. This is also something that I have done before, but I'm very happy with the results that I got today, and I therefore want to show it to you. And I would like to show you first a short video of what I have done. Here it is. Yeah, that's basically me today in the morning. There is this nice plant there, and I've applied some white glue. This is just white wood glue, water-based, and this is how it looks like. Yeah, and then you basically have to wait and notice that the leaf on the bottom side is very, very smooth. That's important. And this is how it looked a few hours later. The wood glue, as you probably know, when it dries, becomes more transparent. So the top one is not quite fully transparent yet, but they're very easy. It's very easy to peel off, also because the leaf is waxy and very smooth. So I've got those little glue flakes here, and see how easily you can pull it off. Yeah. Again, if you want to try that, only use leaves that are very smooth. Now, I have also tried not to use this wood glue, but to use, of course, which glue? The Elmer's PVA glue that can be used also for mounting slides, and that did not work. And the reason why it did not work is the following: do I have it here somewhere? I don't know. Here it is. Here it is. Look. I just want to show it to you. I'm going to move over now to the desk view again. Yeah, look what you've got here. Let me focus here. This is the wood glue, the white one that you just saw. And this one here is Elmer's PVA. And the thing is the following: even I spread it over a larger surface, but it pulled together and made a round droplet. Yeah, so that's not what I want. It has to do again with the surface tension maybe, or the fact that the leaf was waxy. Yeah, it didn't stay apart, so I spread it apart like this here, but then after a couple of minutes, it kind of pulled together, and it didn't even want to stay on the leaf, and some of them actually even dripped off. So that's the Elmer's PVA glue, which is not suitable. Okay, but this one over here, the white wood glue. This is really, so this one over here, that's really nice. And what I have done now is I have basically put the side that was in contact with the leaf, I've put it up to the surface. Okay. Yeah. Yeah, so that's, yeah. Maybe if you put a drop on the leaf and then place a cover glass on the drop, let it dry, and then peel it off with a cover glass, I guess possible. It would be maybe smoother. I never tried that. Yeah, but this system, at least, luckily also works. And I just want to show you now the glue. Okay, so let's just remember there is no biological material on here. It's just the glue, right? Obviously, and we're looking at the glue, and the glue under the microscope. Okay, I have to change over again. Where is the scope view? Here we go. Okay. So that is the that's the Elmer's glue. Yeah, there's some interesting dots here, but it's, yeah. But I want to show you the white glue, and I think this is really nice, because that is the impression of the surface of the leaf, yeah, on the glue. Yeah, so it is just the imprint, correct? Yeah, and what I like so much on this here is that we're even able to see the cells here. Okay, and the question is, are those the stomata? And the answer is, of course, yes. And that's the reason, now you know why I've taken the bottom side of the leaf, because that's where the stomata are located. But something that is really nice here is that, let's go up yet further. Again, no cover glass, no nothing here, right? Is that, yeah, it kind of shows even the individual cells here. This is kind of cool, yeah? So this is just an imprint, and what you've got over here is the left two guard cells, and then in the center a small opening that's able to open and close, the so-called the stomata. And the reason is that this opens up during the day so that oxygen is able to exit and carbon dioxide is able to go in, so for gas exchange. And then at night it closes so that to preserve water. Yeah. So this is something that yeah, I've done this already several times. It always works. Okay, as long as the leaf has to be sufficiently smooth, yeah, then it works. Sometimes if there are hair, some leaves are pretty hairy on the bottom. Not good, right? Because you cannot remove the glue very easily. Yeah, yeah. So yeah. Okay, yeah, and in bright field, okay, bright field, of course, a little bit less colorful. I have to, yeah, but still visible. So that is that's bright field. Now the problem that I have is now, I don't know, this is now 40 times, no cover glass. Okay, bright field, and then let's try the whole thing also using phase contrast as requested. I have to change over, and I have to switch this over, and I don't know how well this actually works. Yeah, this is now phase contrast. Not so good. Yeah, it's a fairly thick specimen, and phase contrast generally works best with thin specimens. Yeah, so that is phase contrast. You can actually see what I think the reason why it looks, the individual cells look a little bit, I don't know, like patches over here. I think this has to do a little bit with the different thickness of the glue, because the different thickness of the glue, of course, also influences the phase shift of the light, which then is converted into brightness differences. Yeah, that's what phase contrast essentially does. It's able to convert transparent objects into darker areas, and that is yeah, I would say an artifact. But if you go back to bright field again, we still we have to move this away, and you still see those patches, but yeah, but they look a little bit like diffraction patterns. Let's try it now. This one over here now, you see how much light it requires. This now is yeah, DIC. I have to make it a little bit. Yeah. Yep. Okay. So this is, let me quickly read a couple of the questions here. Yep. Okay. Yeah. So that's essentially let me go. Sometimes it's better to simply observe them in slightly less. And also the glue is sufficiently flat as well, so that most of them actually are in focus. You see, let's play around with the color a little bit. So quite. I was quite happy with the result. Again, I've tried this already many times before, but I would say this is actually one of the better results. And I think it is really because the leaf was that smooth. So during the removing of the glue, you don't do a lot of damage to it. I've used also different glues, also with solvent. Of course, you don't want to use maybe those too much on plants, but I found the water-based glue works quite nice. So this is just regular wood glue. So let me quickly check again. It's a monocot. That's correct. A little bit of biology. By the way, dicots also have those stomates. Where's the video here again? I'm going to pause now. In biology, what you do is you distinguish between different types of flowering plants, monocots and dicots. And there are plants that essentially have those long, slender leaves. Grass, for example, corn, for example. This plant over here, tulips. Orchids and so on, and the veins, so to say, the vascular tissue runs parallel, so it's not branched. So these are so-called the monocots, right? But you have those the structure of having those stomates on the bottom side is the same for both monocots and dicots. So, and this again, this plant is very suitable because of its waxy leaves, so that the glue does not stick very well, and it's also very smooth, so it can be separated quite easily. Okay, anything else? What time is it? I every now and then I have to check the time. We move on a little bit. The next topic. I just spilled the soy sauce. Here it is. That's always one of those. So that's why we always, luckily the microscope was not affected. Okay, here we go. So this is next thing is the following, also something that I've a couple of years ago done, and what I've got here is this was actually a present of one of my colleagues who visited Norway a couple of years ago, a school exchange program. You know that I'm a teacher, and one of the other teachers visited Norway on this exchange, and she brought this along as a souvenir for me. Nordic elk jerky. So this is dry meat. So this is a dry elk meat, and sold as a souvenir, so to say. And she brought this along, and of course, I said, cool. I'm gonna put it under the microscope, and look what I've done here. Just before the live stream, I added a little bit of water to it to soak it, because I've got one over here as well. Because this elk meat, the dry elk meat, is fairly hard. Okay. This is how it looks like. Looks a little bit like very dry bacon. And of course, this is a way how you're able to store food is by drying it. And why does it store this way? Well, of course, because if something is very dry, then bacteria and fungi have a really difficult time growing on it, and therefore, you can preserve it even without refrigeration. Yeah, so I guess you're salting it, yeah. Yeah, this causes also already an osmotic problem for many microbes, and then you're drying it, and then of course it stores almost indefinitely. And I said, okay, if you, however, want to be able to make a slide with it, then you have to kind of separate and pick it apart, and that's why I put a little bit of this into some water, and we're just gonna make a slide, and we're gonna hope for the best. Yeah. So here we go. And luckily, when I tore off a piece, there was this central piece here, which was very thin. So I just need to get my tools here and let the dissection begin. Yeah, it doesn't really matter. Of course, you can touch it with your fingers as well. And I don't know what this is, but there's this very thin skin-like. You cannot even cut this. Okay. So let's put this also, I would say let's put this skin under the microscope, whatever that is. I have a real problem cutting it. Try different scissors. Yes, this works better. And I don't even know if this actually does contain muscle tissue or not, but it's very convenient because, here, yeah, it's very convenient because it's so extremely thin. So yeah, preparation is fairly easy here. Now I don't remember, did I use what? No, I used alcohol for this tip here, but doesn't matter, it's now evaporated. And sometimes I add a little more water here. And the bottom side is now also wet. I have to also dry wipe it, otherwise it's going to stick on the stage. And really, I have no idea what to expect. Okay, I mean, this meat I have already put under the microscope, but not this thin thing. Here we go. Okay. Yep. You know what? I'm gonna remove the. So that is now the natural. Okay. Well. Yeah, I'm a little bit, how do you say, at a loss. Of course, I know the anatomy of muscle, but this doesn't appear to be muscle. This does not appear to be muscle. It seems to be some kind of, I don't know, maybe some connective tissue. I have no idea. Yeah. How long do I put a plant stem into alcohol? Can you also summarize how to make a plant stem slide? Yes, I can do that. I'll do that later, okay? So I'll do that later, all of the off-topic questions, but this one is definitely not something that I'm super excited about, right? So this is basically the wrong part. Yeah, it was easy to prepare, but actually I wanted to see the individual muscle fibers. Yeah. Okay, let's let's go back to the specimen here. Here we go. So, and now this is where it becomes interesting, because what I have to do now is I have to take an actual piece of the meat, yeah, not just some kind of a weird connective tissue or whatever it might be. And now this actually does seem to be meat. And now what I have to do, I have to really pick it apart. What I have to do now is this meat? Let's see if there are fibers here. The reason is, otherwise it's too thick, and I want to actually see the individual muscle fibers here. This is so tough that I'm also kind of worried that I've caught some tendon or something. This looks like a little bit more like the regular muscle. Yep. Okay, let's. Yes, I know. I should have actually ordered some of those needles. There are those needles, and I've never ordered those. Yeah, maybe because this also kind of works, but this is actually, yeah. This is, you see this part here, this small part? That's actually all that I need. And all of the rest here, I'm just going to put away. So I think one of the issues is that sometimes you don't see anything because it's simply too large and too thick. So sometimes less is more. And this is really little, but we're going to give it a try. I'm checking also occasionally the time. Yeah, I need to. Where is the pipette? I need more. And again, okay. So I hope that I picked it apart enough. Here is one of them. Oh, I bumped it. Yes, barely visible what I wanted to show you. But I need to go up. Yes. This is a microscope test, what I'm showing you right now here. Okay, so I'm going up all the way to 60 times. You can also try this, and if you're able to see that, then you've got a good microscope. And as a matter of fact, even Anthony van Leeuwenhoek, back in the day, the first guy who actually went into microscopy, even he was able to see that. Even with his homemade microscopes. So there is no excuse for us not to see it. That's what I want to show you. This is actually a good microscope test. And yes, indeed, I did find the original publication. Just a second. I did find the actual original publication, and just a second. Not a big problem here. Just here we go. Okay. No, I just had a screen pop up on my computer screen, and yeah. So in one of his publications, he actually wrote about that. He did not know what it is. But if you're able to look very carefully, then maybe you're able to see those stripes. Yeah, you see, these are the muscle fibers, but there are these stripes called striations. They are called, and those striations. Give it the name, it's called striated muscle. And these are called, believe it or not, the light and the dark bands. Yeah, and these are actually protein fibers that are overlapping like this, and they're sliding past each other, and this is how the muscle is able to contract. Yeah, it's a little bit like an accordion, it's able to contract. Yeah, and those striations essentially can be found in all skeletal muscle, and what they found out back in the day, 19th century, some interesting papers. Really, they found well, kind of interesting. If I look at maybe it was Leeuwenhoek himself. If I look at, or was it, another researcher? It doesn't matter. Wow, isn't this interesting? I see those little stripes, and they have. It doesn't matter if I'm looking at the muscle of a whale, the largest animal, or the muscle of a fly, very small animal. Those striations actually all have the same size. This was an interesting realization. The realization is, and that's actually from an understanding of biological systems quite remarkable, is it doesn't matter how big the organism is, on a cellular level, they're very similar. Right, and so this is actually very nice. In my Microbehunter channel, the main channel, some months ago, I actually made a video about this where I talked a little bit about the history of the discovery of this. And I personally consider here very nicely visible. Here, look, the striations here are very nicely visible at the light and the dark bands. The striations are also called sarcomeres. Not quite. The sarcomeres is the individual contractile unit of the muscle. The striations are simply the alternation of the light and the dark band. Yeah. So basically because of the sarcomeres, you have the striations. I know this is kind of a little bit, how do you say, maybe getting a little bit, too detailed here. But there is the sarcomere is actually one contractile unit, which contains the light and the dark band, and they basically make these stripes. Striations is a fancy way of saying stripes. Yeah. I have done this with other meat as well. Cooked chicken meat works as well. So yeah, so this is I think a nice microscope test, so to say. Yeah. When, yes, it's a little this hobby of microscopy and so on. I admit I'm a little bit. I like being a little bit geeky here, but then I have to say the following. The whole thing with the striations and with the striated muscles and the light and the dark bands, actually this is stuff that I have to teach in school. That's what stuff that my students actually have to know. Yeah. So it is strictly speaking, it's not even that geekish or geeky or nerdish. It's part of the curriculum that I have to teach. Yeah. So just but one of the things that I personally consider interesting is not only the biological details of how this actually happens, how when the thick and the thin filaments, as they're called, when they slide past each other, and how the muscle contract. All of this biological detail is not so interesting for me anymore. I'm more interested in the actual reasoning that went on in the people who discovered this, because I think it was Leeuwenhoek who saw those striations, those stripes first. He didn't know what they are, obviously not. Yeah, thank you very much. The questions you're here, but he actually thought that this looks like a wire wrapped around some kind of a central core. That's kind of what he compared it to. Right. And then only later they discovered that that's actually not something wrapped around in a spar, but actually these are indeed stripes. Yeah. Yeah. Where do I teach? I teach in Austria, in Europe. Yeah. I'm a regular biology teacher, so to say. Yeah. So, but I teach in English. Yeah, so in our curriculum we're teaching in English. Yeah, so this is simply something that I just wanted to show you, because sometimes I like revisiting those samples that I've already looked at before. Let's go down a little bit here. Yeah. Yeah, and you have to see if there are too many of these muscle fibers on top of each other, we cannot see those quite well, so you have to be a bit patient. And here, where is it again? Yeah, here on the side we see them. Yeah. So I'm gonna move on. These stripes are not refraction differences on density. No, they're not. They are because I wouldn't say refraction differences, but actually these are where there is more. There's this protein called myosin, also known as the thick filaments, and they basically show up as the dark bands, so to say. So this seems to be indeed, I would say, probably a denser area, which probably does not allow light to go through as much. Because these are not these are not actually. You know what? Let's see in phase contrast. I don't know what phase contrast tells us here. I only have 40 times phase contrast. Yeah. Yeah, it's not such a huge difference really. Yeah. Not such a huge difference. And in bright field, let's have a look in bright field again. 60 times. This is now like this, and that's now bright field. Actually, in bright field, we see it quite well here, right? And I've opened the condenser right now, so the effect of diffraction should be quite low. If you close it, then the contrast goes up again. Yeah, looks quite nice. Yeah. Of course, one possibility would now be if I could use a micrometer slide, I could actually measure out the distances. Yeah, and depending on whether the muscle is contracted or relaxed, the width of those light bands, yeah, the white areas, they basically become wider when the muscle is relaxed and smaller when the muscle is contracted. Yeah, so you can actually, by the width of the light and the light bands, you can actually determine of whether it's contracted or relaxed. You see this way better, of course, if you use an electron microscope, yeah, which we obviously don't have. Yeah. So you know what? I move on. Last part. What time is it? Last part. No, not last part. Density gradient separation. Wow. Okay. Yeah. Bright field wins today on this one here, and DIC wins on the crystal. Yeah, that's correct. There you see, there is not one imaging technique that you can say is automatically better than the others, right? Because if you use DIC for over here, now that's DIC, right? Yeah, it requires a lot of. Yeah, first of all, it doesn't look so much better, right? This is DIC. I have to. Yeah, it doesn't look so much better. Yeah, I can change the color here a little bit, but yeah. It also introduces fake colors, and that's also something that you don't always want, right? So you see, that's why, that's why there are so many different techniques out there, simply because for different specimens and depending on what you actually want to see, right? For bacteria, for example, phase contrast is really cool because you see them much better. But for thicker specimens, phase contrast does not. It just looks more blurry. Yeah, so okay, the next thing is yeah, if it's getting too late. No, it's not getting too late. I just thought that I just want to try this in any case. It's not so much of a microscopy thing, a little bit maybe, but I wanted to try this. I tried it today and it didn't work on the sample, so I said I'm gonna do a test sample. So look, I'm not gonna use that. This here is some rainwater that I collected. Okay, a few days ago. It's been raining here in Europe like crazy over the last 3 weeks. Finally, the weather's getting better. Now it's getting too hot. You cannot make anyone. Doesn't matter if it's when it's raining, people complain. I complain. When it's getting too hot, people complain. Yeah, okay, doesn't matter. We always complain. What I want to do is the following, and I need to tell you a little bit of why I want to test this out. Here's the desk view. Yeah. Rain in Europe, no way. Ever small. I think you're in Britain, right? I mean, it's even more rainy than here. So density gradient separation. So the thing is the following. And this is why I need the salt water again. That's actually why I made it. I've read somewhere that in rainwater, they found microplastics. The question is now how do you get the microplastics out of the rainwater? I tried it with rainwater. I was not able to find anything which is worth showing. Okay, so I'm not going to use rainwater, but I'm going to use this water over here, which I made. I'm going to explain what it is. And in order to separate microplastics out, there is a common technique that you use, and that is you take a tube, you add the water. Okay, you add the water and you add salt water, concentrated salt water, approximately the same amount of saturated salt water. And then you have to let it stand, and I don't know if this is going to work today because of the waiting time and so on, you get the idea, but I want to try it in any case. Because when you add salt water, what's going to happen is that the lighter microplastics, or not the lighter ones, the less dense microplastics, which basically had the same, will start to float on the surface of the water. That's the idea. It's a little bit like I don't know, maybe you've heard that if you're in the Dead Sea, which is saturated salt water, people can just float on the water without swimming because of the density of the water. And we want to do something similar here as well by actually adding salt water to a sample. We want to kind of force all of the less dense particles up to the surface for investigation. That's the idea. And in the rainwater, this was a little too clean for my purpose, and for this reason, what I have done here is I just made a test sample, and I simply took some dust from my computer. really, on the surface of my computer, mix it with water, and that's what we got here. This is kind of dust water. Okay? And I want to see now if indeed this technique is able to force some of the dust particles, whatever it might be, maybe some fibers from clothing, some microplastics from textile, I have no idea. Maybe I want to force it to the top, and I don't know if it works. Okay? So this is, something I would like to, yeah? My face camera has a little bit too much exposure. Yes, I've seen this, yeah? I think it's not the face camera that's the problem. I think the ambient light that I have. Yeah, I'm just gonna switch this off. Okay, I've got an extra additional light here. So, but let's try this. Let's go a little bit higher here, and I'm going to simply add approximately 7 mils of this dust water. And I don't even know if we have enough time on how long it takes for the separation to happen, if separation is possible at all. And I'm also going to directly put this under the microscope as well. So same amount of saturated salt water. Any salt crystals would now dissolve as well because, And now you have to shake this, and now you have to kind of wait and hope that some of the particles will start to go up. And I'm just going to put it somewhere. I'm going to wait a little bit. And in the meantime, I'm going to answer some of the questions, or we're just going to put some sample directly under the microscope. And this is basically a test, because if this works, then I can use this method also for other water samples to kind of separate certain things. However, what they usually do, what I heard is for microplastics, is that you normally want to use it, pass it through a filter, because simply you're able to process a larger volume of water. Okay, so this is kind of the dust water. And let's have a look. I need a cover glass again. Yeah. And we take out the meat, the elk meat, and let's have a look at. You always start at the lower magnification first, as always, and where is the scope view? Yeah. So you see, it's 4 times. Yeah, you of course, as expected. Okay, as expected, lots of fibers, very often textile fibers. Those things that you see here, I think you see them quite common. I think these might be some parts of a plant. Those here, yeah. Not sure which one, but I think these might also be some plant here. You find this quite frequently in dust. Photographic CPL, circularized polarizing filters, could be used instead of linear polarized sheets. Yes, I have done so before, but be careful, because if you use circular polarizing, then also the orientation is important. Yeah, so you have to try it out first, the correct orientation, that you have complete, darkness. Yeah, because the circular polarized filters have also a lambda fourth plate on one side, so this means the orientation like this is also important. Okay, side issue. Yeah, so this is the hair of a carpet could also be a half. No, it's good point. It's not the hair of a carpet beetle. Because I actually have found here of a carpet beetle, which looks a little bit different, similar but a little bit. The structures are slightly finer. I think could be, but I've got some permanent microscope slides where I was not sure what it was, and then many years ago I posted a question in a German microscopy forum, and then actually they identified it. And there is, unfortunately, I do not know the English term, but in German, for those of you who speak German, it's called Pfeilhaar, arrow hair, so to say. Yeah, So I've seen some plants also have similar ones, but I have to admit that for plants this is indeed a little bit too fine. Yeah, but indeed certain larvae have those setae, which are essentially here. I don't know about this one here, right? But for the carpet beetle, I've actually found one, and they have a very interesting structure. Yeah, carpet beetles actually are not not a nice thing to have, because of course they eat carpets. Yeah. So, yeah, you see, that's basically these for your info. Yeah, that's actually because microplastics are basically now in the media quite a bit, and one main source, or not main source, but one important source of microplastics are actually synthetic fibers from clothing, which are released into the environment by the washing process. Yeah, and what we can see, of course, is this is dust that has settled on my computer. Okay. Yeah, so, and this dust that has settled on the computer, you can see there are lots of fibers here, right? So, and what I'm going to kind of do now is with this here, is I'm just going to wait a little bit more, and yeah, maybe until tomorrow, and then maybe I'm gonna report back on, or next week, on whether, or I'm just gonna let, or I'm gonna let it stand for 2 weeks now, and to see if indeed the fibers, which are less dense, have actually accumulated here at the top. Yeah. Yeah. Do carpet textiles have nutrition? Cellulose. That's all I can say. Yeah, clothing is, yeah, moths, for example, they also eat clothing. Indeed, certain insects are able to digest cellulose. Yeah, cotton is cellulose, and that's an energy source. Yeah, indeed. Yeah. Not only cellulose, but I don't know, all sorts of substances. Yeah, nature is very how do you say? If there is an organic substance in nature somewhere, chances are good that some organism is able to eat it. Yeah. Can we quickly see last week's mosey, please? May I ask, what's a mosey? Are you referring to the insects, the mosquito? Which is not a mosquito, as I found out. Yeah, I'll quickly show you indeed that what we mounted last week. Mosquito. It's not a mosquito. It seems to be some kind of a fly. I forgot the name. Yeah, and the reason is because of the mouth parts, and because it does not have any scales. Yeah, so it's not a. It's of course related to those, but it's not a. Yeah, I don't know. Yeah, but it's not. Unfortunately, the clearing process worked on the legs, but not so much on the other parts. Let me quickly go down here a little bit. Yeah, a midge. Yeah, yeah, exactly. So this one here is one of them. Look at the dense antenna, and the other one. There's another one. You know what? I'm gonna go. Let's do the lowest magnification here. Who cares? The lowest. So a little bit of dust is also in here, even if it's not so nice to look at. We get here. Yeah, you can actually see that this one over here, the antenna of this one over here is different. I don't know. Maybe the one with a denser antenna is a male, the other one female. I don't know. I don't even know if they're of the same species. But the first thing that I realized is it's not a mosquito. It's just look at the mouth parts. Look at the mouth parts. Yeah, this is not a one that's designed for sucking blood. Yeah. And also in a mosquito, the wings, what they have is they have those scales. But what we have over here is this kind of hair-like structures. Yeah. Which is also a little different. Yeah. So just, and I kind of hope, because I used U-Pearl to mount this, and normally after some time the insect will become more and more transparent because of the clearing action. And what I'm, however, a little bit concerned about, and I also made a video of this, is, dang, I did not put those insects, maybe long enough into alcohol. Yeah. Because maybe there is still some air in the center somehow. So I don't know. Yeah. I did put it a little bit into alcohol, but it has to properly infiltrate the whole organism. Yeah. Yeah. Again over here. Yeah. I think just a second, there was a third one. 1, 2, where's the third one? I don't know. This one here. That actually is quite transparent. This is the rest of the when it undergoes metamorphosis, when the larva becomes the adult, and this is kind of the adult emerges out of that. It's like the skin, yeah. Yeah. So let's do the following. Let's quickly have a look at some of your questions. Yeah, just very quickly. I'm going to jump over those that are already answered, okay? Let me see desk here. Here it is. Okay, I gotta remove all of the wet microscope slides here. Otherwise I'm gonna get the paper wet. Yep, here we go. Yeah, see, that's basically, some of these are the comments from the spider at the beginning. It's UV or shortwave length light. So some of these are just comments here. Okay. What warmer is that, and did you get it from Amazon? Yes, I got it from Amazon. It is a coffee warmer, and it is extremely practical, and I actually used it already several times. However, there's one recommendation that I have. I got one, I can use it here, I got one with a power connector. Many or most coffee warmers are USB, and the USB will not give you sufficient power. So for this is the reason why I got this here. It's indeed, there is this rubber pad on top here. You can adjust over here the temperature. I've used it also in previous videos to melt the mounting medium, the glycerin jelly. Yeah. It goes pretty hot here. There is also a timer function here, which which you don't need. Yeah, 3 hours, 4 hours. It's yeah. Not needed at all, but yeah. Otherwise, very useful. Yeah. Yes, I got it from Amazon, and if you get one, I recommend that you get one with an actual power supply. Yeah, because you simply get more power. Maybe use high magnification. Let's move this up here. Let's maybe use high magnification with cover glass to induce crystallization. I talked about this already. Can we do this in the same effect but quicker by using alcohol? We tried that and it didn't work. It's something interesting that we found out. Evaporation time depends largely on air movement and less on the temperature. You can use a hairdryer to observe the process more quickly under a microscope. The thing is, if you have air movement, what you're doing is you're carrying away the air molecules. So that's... I would say, look, I think it's a combination of multiple things. It's also a question of ambient moisture that you have, or humidity that you have. And if you have got temperature, surrounding temperature, of course, then you're able to speed up the process. If for example, you have air with a lot of moisture, for example, and little air movement. So it's, I would say, probably a combination of all. But it's true. If you want to dry your clothing and in a small room, which has already 100% moisture, it's not going to dry, right? Then you need air. You need fresh air to carry the moisture away. Yeah, I think that's very clear. That's what's meant here, right? I looked at the seawater under the microscope. It happened way faster than I expected. I wanted to do the time lapse, but it happened rapidly in front of my eyes in a matter of seconds with a cover glass. Okay. Yeah. So, yeah, basically crystallization happening very quickly. So any substance here would be evaporates, yes, from a mineral. So any substance in here would be. Well, no, let's put it this way. I guess if you want to make crystals, then what you want is you want to have, not all substances crystallize, obviously, right? And they have to be dissolved as well. And when it evaporates, it has to, go into a crystalline state and not stay amorphous. Yeah, so, and sometimes it also depends on the speed. Sometimes some substances take a longer time, and if you evaporate it too quickly, then the crystals might also not form. Yeah, So there are a variety of parameters here, depending on the substance, on the solvent, and also on the environmental conditions. Yeah. I wonder what would happen if you put a drop of soy sauce into alcohol. Yeah, we tried this. Okay. How do you determine if a substance can make crystals or not? How do you determine it? Well, it's got to have a it has to be able to form certain bonds with each other in the absence of a solvent. Yeah, and I mean there are certain chemical characteristics certainly that determine that, but it depends also a lot on the solvent that you use in other conditions. Yeah. Beta hydrochloric acid. I don't know so much about that. Okay, so we have to see if this actually works. Yeah. Maybe if you put a drop on the leaf and then place a cover glass on the drop, let it dry and then peel it off with a cover glass. Yeah, that's basically trying to get the glue on the cover glass, and so that it's easier to peel off. Yes, there are stomates. We tried bright field and phase. Dark field I only have it in 4 times. That's a little bit the problem. So I'm not able to see a lot. Yeah. I wonder if the resolution of the imprint would improve if you let the glue dry in a vacuum chamber. The thing is the following. I think it's a practical problem. If the glue has to dry on the leaf, right? And if I want to dry the glue in a vacuum chamber, I would have to put the whole plant into the vacuum chamber. And I think the plant might not like that because it's actually going to dehydrate the plant. Yeah? So that's a little bit, I would say that's a little bit maybe of a practical issue here. Yeah. Off topic, what is the plant stem you have in alcohol that you microtome with solid core? And how can one specifically hunt for solid core stems to collect? What is the plant stem you have in alcohol that you microtome with the solid core? You mean the stem that has a solid core? And how can one specifically hunt for solid core stems to collect? I don't know which one you are referring to right now. Generally, what I would recommend if you want to try cross sections of plant stems, then do the following: get non-woody plants. So, for example, you just go into a flower shop, buy one nice large flower, a tulip, for example, because they often have very thick stems, and also non-woody stems. Why non-woody? Because they're much, much easier to cut. And what I've done then is I chopped up the stem using a sharp blade into sections like this and placed them directly into concentrated alcohol. And this will actually remove the green pigment, the chlorophyll. Yeah, and that's much easier to collect. So my suggestion is I mean, that's what I've done is those nice plant stems that I've cut easily actually are those of flowers. Yeah, one of them was a tulip, I think, and the other one the sword lily. And I basically put those plant stems cut it into sections. And then simply dropped it into alcohol, and they've been there for many years already. Yeah? The plant I found, sadly, is 90% hollow. Ah, okay. Okay. You mean basically it has a hollow stem. Yeah, if it's hollow, then like, for example, dandelions, they have a hollow stem. Difficult to cut. Difficult to cut. In the end, could you please have a quick peek at your mosquito? Yes, we've just had that. Yeah. How long do you put a plant stem into alcohol? Can you also summarize how to make plant stems slide? You know what? Let me just give me a minute, because this question reappeared over and over again. Give me a minute. Here it is. Okay, finally, luckily found it. Yeah, so these are basically plant stems, non-woody plant stems, and I basically put them into alcohol, and you can keep them in there. I have kept some plant stems in concentrated alcohol since I started microscopy, over 25 years, really. They can store in alcohol practically indefinitely. You can see that the alcohol will turn greenish after some time. Then you pour it off and you add more alcohol until it stays clear, right? And then what you do is you take out one of those, yeah, stems. Actually, I did make some videos about this some time ago, and then you use a very either a microtome or you use just your fingers and a very sharp razor blade, which I don't know where I put. Okay, yeah, I don't know where I put it. And then you try to make very thin slices. What you can also try to do is if you cannot get it very thin, you can actually cut diagonally, because there where the blade, it will become very thin. Huh? So that is, just get yourself a sharp razor blade, and once you've put it into alcohol, you're actually going to feel that it will actually turn a little bit harder. Yeah. Okay. There's a question about the tubes that I have here, which I quickly answer. Looks like the membrane between the skin and the muscle. Could you also try to cross-polarized light in some of these crystals? I don't have a standard cross-polarized, unfortunately. I have basically a Waterstone prism in there. Okay, and so this means even though I have polarized light, I don't have the standard cross-polarized. So you see, no microscope is perfect. Yeah. So very quickly, do those tubes you have a rubber sealing ring in the lid? No, they don't. Okay. So basically, they don't. So these are actually cheap centrifugation tubes, and they are indeed tight, right? Even without a rubber seal. Okay. So that's watertight, and also here I've got another one. Those here as well. Okay. So yeah, they're quite good in that respect, because they're very versatile, and I've got a large number of those. Yeah. Yes, it works without the seal. As a matter of fact, a short story. Back in my lab days, what happened was luckily not to me. There was some liquid in there, some water, and somebody put it into the microwave to heat it up. But the person forgot to unscrew it. You have to unscrew it a little bit to allow the vapor to escape. And the person did not unscrew it, but had it tight. And then it exploded. And how it exploded was quite interesting, because there was actually a hole on the side of the tube, because the steam pressure was so high that it exploded. So this means that a cap was actually stronger on the container than the actual wall of the container. Just saying, right? So it was quite interesting. Yeah? So it did not blow the cap off, but the side of the container exploded. So it kind of shows how strong the cap is. Yeah? These tubes are microwavable. I see no reason why not. If you put liquid in it, it's going to become hot. The plastic's going to become soft. I never microwave them. But I can imagine that you should be able to, because you have to, maybe sometimes you have to autoclave them and you have to sterilize them. Something to try out. Yeah, but always don't put liquid in it and put a lid on top. Yeah. So and you have a little firework in the microwave oven. Yeah. So let me quickly go through the comments, if there are any comments left, and otherwise I'm just gonna call it a day today. Yeah. Yeah, the carpet beetle question. Like the hair, maybe it is the hair of a carpet beetle. I don't know. I have to make a check with one of my old slides. Yeah, face camera with too much exposure. Again, look, even though I turned off the light, the exposure is still too much. Interesting. Yeah, you see that it's a little bit overexposed. There is a possibility to change the intensity of the light. Maybe that's better. Yeah. Okay, I think there are no more comments over here. Yeah. Yeah. So I'm gonna call it a day again. Today, Yeah, I wish you all the best. I hope that you enjoyed this week's session. It's always one of the reasons why I do those live streams. Is not only, I have to be honest, not only for you and for the community, also for myself, because I'm having a great time here as well. Obviously, yeah. So, and those me making YouTube videos and doing live streams and doing stuff like this is, of course, it's a hobby. It's a little bit also a way of life, maybe for me. Yeah, simply going around into nature, looking around for specimens, and I'll reveal something about me. Yeah, I'm a biology teacher, but you see, sometimes when we teach biology in a classroom, it can become quite theoretical as well, and those practical things, sessions, actually keep me grounded a little bit in that sense. And otherwise, there is always a little bit the danger that one loses connection and contact with nature and the actual real world. Yeah? And that's what I kind of also want to prevent a little bit. And plenty of things to play around with in any case. I wish you all the best. Please do have a look at also at my other videos of this channel. I've been uploading one video almost every day now for about 2 weeks. I won't be able to maintain that density of videos in the future, obviously, because when the holidays are over, I have to go again to work, and I'll fall back to a slightly less frequent upload schedule. But have a look at those as well. And if you have any comments, and that's really important, I've been thinking a little bit into which direction should I take the channel. I'm now trying to separate the microscopy channel from the microbiology channel a little bit better to have more targeted viewers. But if you have any also critical comment on what to improve, I don't know, maybe I'm talking too fast or too slow, or I don't know, right? Maybe I'm rambling around too much. Yeah, so it's fine. Any form of feedback is welcome. For me, it's after all, something that I also would like to improve on. I'm gonna call it a day. Will I have live stream next week? No, not next week. Okay. Again, if you joined in late, I will not have a live stream next week because I'm not here, not around. I will try. I don't know how good the internet connection is. Maybe I'm going to keep you updated a little bit by community posts. So there is the possibility to post pictures and text. So check that on the channel. Maybe I'm able to upload a couple of short videos. I don't know yet. Depends a little bit on the internet connection and whether my laptop computer is fast enough for video editing. Enough for me. From me today. All the best. Happy microbe hunting. See you around. Bye-bye.